Hi everyone, I'm Florencia Gottardo. I want to talk about exploring the metastatic role of inhibitor of apoptosis Bergna in breast cancer. This work was made in el Instituto de Biotecnología y Biología Molecular de la Universidad de La Plata, Argentina, and Laboratorio de Oncología Molecular de la Universidad Nacional de Quilmes, Argentina. Breast cancer is the most common cancer and the first cause of death in cancer in women worldwide. Bergna is a member of the inhibitor of apoptosis protein family, thought to play an essential role in progression and tumor resistance in many cancers. The aim of the present work was to investigate the role of apoptosis inhibitor Bergna in breast cancer, focusing particularly on its involvement in metastatic cascade. To evaluate the role of Bergna in human breast cancer samples, we proceed to study transcriptomic databases. The expression of Bergna in samples from primary tumor in breast cancer was higher than patient tissue samples from healthy voluntaries. Also, breast cancer patient survival was evaluated using data from TCGA Pan-Cancer Atlas. Patients were divided into groups, high and low Bergna expression, and survival time was significantly lower for patients with a higher Bergna expression levels. When we evaluated Bergna gene copy number variation, we observed a direct relationship between copy number and NRN expression. Samples with gene amplification have the highest expression levels. We decided to investigate the relationship between CMV and different clinical attributions. In this graph, we observe a higher proportion of amplification and gains of CMV in negative estrogen and progesterone receptors. And we assess the influence of CMV on two parameters that reflect prognosis, the Nottingham Prognostic Index and histological neoplastic grade. And patients with amplifications and gains have a higher Nottingham Index and histological grade 3. We also observed that patients with a higher CMV tend to be diagnostic at young age. And we evaluated Bergna CMV distribution according to chemotherapy and radiotherapy treatment. And the figure showed that there was a greater amplification and gain percentage in samples for patients who received either of the two therapies compared to those who did not. For more systematic approach, we conduct a differential expression analysis followed by gene ontology study. Differential genes obtained in this analysis were divided into two output groups, overexpressed and less expressed genes, both under condition of high Bergna expression levels. These figures summarize biology process, cellular components, molecular functions, and pathways involved in overexpressed genes. We analyzed the correlation between expression level of Bergna and different proteins involved in apoptotic. The results show that those samples with high expression of Bergna had lower expression of pro-apoptotic proteins like BAX and higher expression anti-apoptotic BLC. Also, we showed that samples with a high expression of Bergna had increased expression of some proteins that promote proliferative pathways. Furthermore, we evaluated the correlation between Bergna overexpression and critical regulation of metastatic cascade, particularly in angiogenesis, migration, and adhesion proteins. We found that those samples with a high expression of Bergna had high expression levels for some angiogenesis promoters such as HIF1 and VEGF, and higher expression of proteins that promote migration are involved in focal adhesions. So our conclusion is genomic results show that the higher copy number of Bergna genes correlate with a more aggressive phenotype and a worse prognostic marker. Transcriptomic studies show that BRCA is overexpressed in tumor cells and correlate with an anti-apoptotic proliferative profile. BRCA could play a role in the activation of signaling pathway involved metastasis, and the result of in silico analysis present here make it possible to postulate the inhibitor of apoptosis BRCA as an interesting molecular target for the development of a specific therapy against breast cancer.